An important non-specific defense against tissue damage is the inflammatory response. The inflammatory response commences when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Adhesion molecules known as selectins are displayed on the activated endothelial cells within the capillaries. These selectins attract neutrophils, slow them down, and cause them to roll along the endothelium. The neutrophils encounter chemicals that will activate adhesion receptors on their surfaces called integrins. These integrins then tightly attach to the adhesion receptor molecules on the endothelial cells, causing the neutrophils to stop rolling and stick to the endothelium. Neutrophils accumulating along the walls of the capillary is termed as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. The release of histamine stimulates vasodilation as well as the opening of the junctions between the endothelial cells, which allow fluid and leukocytes to leave the capillary and enter the infected tissue. Neutrophils squeeze through the endothelial wall into the interstitial tissue fluid by undergoing dramatic changes in shape in a process called extravasation. Chemotactic substances released by bacteria and tissue breakdown products attract neutrophils and other types of phagocytes, which will ingest and destroy invading bacteria.
The signs of inflammation: dolor, calor, rubor, tumor, and functionalisa, which means pain, heat, redness, swelling, and loss of function respectively. The cytoplasm has four distinct types of granules. First is primary or azorophilic, secondary, tertiary or gelatinous, and lastly the secretory vesicle. The primary granules. These contain myeloperoxidase, which converts hydrogen peroxide into hypochlorous acid, which reduces the pH and initiates the destruction of the bacterial cell wall. Lysozyme is an enzyme that disrupts the structural integrity of the bacterial cell walls by breaking polymeric beta-1,4 linkages. The secondary granules. These contain additional enzymes such as apolactoferrin and collagenase, which prevent bacterial growth and increase the PMN mobility. Apolactoferrin binds free iron and prevents the bacterial synthesis of heme-containing proteins. The tertiary granules. Tertiary granules contain lysozyme and gelatinase. Lysozyme breaks beta-1,4 linkages, while gelatinase degrades ground substances between cells and increases PMN mobility. The secretory vesicles. These secrete N-formyl, 1-methionine, 1-leucil, 1-phenylalanine, which is a chemoattractant and an activating agent for PMNs. Those are the polymorphonuclear neutrophils.